Okay. Um, we are glad to have Dr. Shivendu Ranjan as today's speaker. Shivendu is an assistant professor at Indian Institute of Technology, Kharagpur, and has expertise in nanomedicine, biomaterials, nanotoxicology, and nanonutraceuticals. Shivendu did his PhD from Vellore Institute of Technology, and after his PhD, he served many organizations like Indian Institute of Food Processing and Technology, a Government of India organization, Eastpin Nanotech at IIT Kanpur, University of Johannesburg, South Africa, and um, lastly, Gujarat Biotechnology Research Center, DST Center, before he joined his permanent faculty position at IIT Kharagpur. Shivendu is a life fellow of many reputed scientific societies, including Indian Chemical Society, the Linen Society of London, Indian Engineering Teachers Association, and Bose Science Society. He is also an associate editor of a very high impact journal, Environmental Chemistry Letters, published by Springer. Shivendu has a very strong and scholarly scientific credentials that is evident from his 55 research papers so far, and he has edited 34 books. I have to learn how he manages to do that. And he himself authored 2 books. He has several patents and 4 prototypes for industries. So, with this brief background, let me welcome Shivendu to deliver his talk, A Biomaterial Engineer's Viewpoint on Bone and Joint Research. Shivendu, the
Yeah,
floor is all yours.
yeah, thank you so much, sir. And thank you for the great initiative that is Vasti Center and regarding this, the talk series, which is which actually includes the interdisciplinary, thought process. And as I told, since I'm on holiday, so I have a special student with me. She is my niece. So let us start the talk now. As Sir has told that biomaterial engineers' viewpoint on bone and joint research. Let us understand the biomaterial and engineering perspective with respect to the bone and joint research. So, before going to the deep into recently I joined IIT Kharagpur, and at that I started the nanobio research lab. I started stabilizing it, and I started my work with the drug delivery and polymers, a bit of skin and cartilage challenges to be solved in collaboration with the I'm still learning with Navidya sir, and now we are slowly entering into the bone and other portions of the biomaterials. So, as sir has mentioned, I'm a complete bioengineer. I have a background of formulation materials perspective, entering into the different domain of a bone, I face a lot of challenges to understand how to inculcate my engineering perspective in that. So today's talk will be focused on that, on the very the to develop the interdisciplinary thought process on incorporating the materials perspective in bone and joint research. So before going, so I would like to again, like all the scientists, I would like to showcase the products which have been in which I have been involved. As Sir has told that I my area of interest is nanomedicine, that drug delivery, antimicrobials, and other stuff, and toxicology. So this is how I started my journey. Bioengineers with a special reference of materials. It's a tough journey because understanding the biology, understanding the materials, understanding the biology and materials together it was a tough. So till now, the products what I have developed are so few of the vitamin delivery through oral thin films, face mask. These face mask is quite very common in mainly in UP because the manufacturers are from UP that is based in IIT Kanpur. A nozzle, this I'll be describing in the next slide also, and some of the formulations on wound healing. So this nozzle, I like it has been patented. The patent is published in WIPO and USPTO. So this nozzle is actually to enhance the process of electrospinning through only one outlet. We can have multiple pores and we can create multiple jet of the polymer to spun it into the fibers. So from a single outlet, we can create multiple jets. It has enhanced the process in multiple folds. Using the electrospinning, I have lab scale prototyped the face mask using the pan PSX blend and different formulations. This has been further scaled up by the industry and it has been tested by Nelson Lab, CE, ISI, including SITRA and ISO in India. And the face mask is being in market from many years. This is the vitamin D3 through oral films. And again, on this, over the placebo, we have incorporated the drug, encapsulated drug, to deliver it quickly as well as efficiently through oral route. This formulation have been involved in prototyping for Dr. Reddy's. Using the edible protein, we have encapsulated the drug into the edible protein and again electrosprayed or spray dried it to bring the size as down as possible, even to the nano sized. And the formulation should be of uniform size, uniform drug distribution. The major challenge of this encapsulation was to develop a drug mainly for kids because kids cannot be able to swallow tablets and capsules. This is the comparison, normal comparison, that spray dried and electrosprayed. So obviously in electrospray, we can have uniformity into the spares and uniformity into the drug loading. Uniformity means the monodispersity is quite possible in electrospraying rather than the spray drying. Again, this is a hernia mesh on which we have coated the antimicrobial means antibiotics, and that too on the single side, so that after the post-operative condition contamination can be avoided. Some formulations in wound healing with different in collaboration with different companies, that is Environ Care Products and AP1 Labs. Again, some formulations of vitamin delivery. So coming directly into the rather than showing all the formulations, let us jump into the bioengineering part. So this is a summary of the products. So let us this will again take 2 hours to 3 hours to describe all those stuff. Now, as I can see, the audience are majorly from the student background. So I have kept it as simple as possible and as critical as possible to generate a critical thought process into the scientist fraternity, mainly the young scientists. If we consider the bioengineer, usually bioengineer term is actually not very clear among the bioengineers as well. If we focus on the bioengineering terminology, we first think about the genetic engineering, but the bioengineering overall terminology is not only the genetic engineering. It should include the knowledge of mechanical engineering. It should include the knowledge of material science engineering, chemistry, physics, chemical, mathematics, and nowadays, once the era of the machine learning and artificial intelligence, the computer science and electrical engineering as well. So bioengineering overall is a very bigger term, very interdisciplinary term, and in that, again, biomaterials. What we actually understand from the bioscience means biomaterials science and engineering. If we consider the biomaterials, actually the biomaterials for every part, it should be with respect to the site of the function. Maybe for the bone, we need different biomaterials. In bone, again, for the soft bone, for the for different types of bones, we need different biomaterials. If you need for the vascular biomaterials, that should be different. Skin graft, that should be different. So biomaterials are the synthetic or even natural sometimes. It should be in the contact or should interact with the biological system. It means even a wound healing patch or a drug delivery vehicle, these are the biomaterials which has the biological functions. It interacts with the biological system. They are in contact with the biological system. And if we consider biomaterial as a whole, so the mantra which I realized after working for different industries and different labs, biomaterial selections are depending upon different site, different functions, where actually we want to implement what application we need. At our right hand side, we can able to see we should have different formulations or different formulations has been identified. If we should have the dental implant, if we should have the biomaterials for ear, heart, prosthetic joints at different levels. So it depends upon the site, functions, applications, and even microenvironment. Why I am focusing on the microenvironment? Because recently, while in a discussion with Navidya sir itself, suppose we are developing a biomaterial for a person who is already overloaded with the infections and microbes, then we should actually either encapsulate or coat our biomaterial, we should contain the antibiotics. Otherwise, the chances of contamination, slow degradation, and many other challenges on those biomaterials will be possible. So we should actually not ignore the microenvironment of the site. Again, to have a broader perspective of the biomaterial, the bileaflet heart valves and the dental implants, as well as the vascular grafts, these are not the same biomaterial, or the same biomaterials concept cannot be applied to the bone. Means all these three. Till now, I'm not discussing the bone, we will slowly come. Let us say if we are developing the vascular graft material should be flexible enough. It should be designed with the open porous structure. It should it should not it should not get recognized as a foreign body. It should be biocompatible. It should actually work as how our vasculature structure works in our body. But in case of dental implant, that's different. That can be the metallic, or that can be biocement. If we consider bone, even bone is more challenging. In our screen, we have femur. The structure of the proximal part and the distal part are different. Even the structure of the middle portion is different. And even in the proximal part, the mechanical property, the biophysical property of different portions of the proximal part is different. And we need to understand it very critically to develop the biomaterial. Same for the distal part. And obviously, like as I told, being a bioengineer, I should focus on the material perspective, means one should focus on the material perspective as well as the biological perspective, as well as the physics perspective, the chemistry perspective. So we should develop a global scenario of the biomaterial to understand it or to hit the unit precisely. If we consider the hip implant or total hip replacement, the hip implant was first attempted in 1800s. If you refer the book, and it has grown. Now approximately 10 lakh total hip replacements per year is being reported by several articles. But still the challenges in the hip replacement. Major challenge is the infection. Whether our body is accepting the biomaterial as a biocompatible or not, whether the immunal responses are coming over or not. So many challenges are there. Even the upper part of the femur, the distal part of the femur, the middle portion of the femur. So these all are different, and we should developing let us say if we are developing the femur, artificial femur, as a single unit. So in that again we have to conjugate, or we have to think about development of let us talk about just the compactness. So proximal compactness and the distal compactness are different. How can we meet? And if we do meet, means if we will try to develop these as a Lego perspective, then joining them, giving the mechanical strength to this femur so that it will bear the load of the person, it will not crack in between, it will sustain. So these are the challenges. So how to develop this? Let's have a look. You have two parts, part one and part two. Part one is actually discussing the challenges of the stem. If we should actually, we are bound to develop three different material to cover the proximal part in hip replacement. Similarly, the material challenges are in part two for the femoral head. It should be stable enough. I'll not talk about the critical because as per the information received from Swati, the major portions are the students. So instead of going to the critical and mathematical perspective, in a very simple layman perspective, we should develop the material which will sustain. With respect to it, it should have the it should be similar to the whatever the biomaterial or whatever the portion, if we are developing the proximal or distal. In every aspect, our body should recognize it as their own. It means it should not have rejections. So if we consider in these two case studies, the major challenges are how to develop this. Because we should consider the stem, we should consider the cup, we should consider the plastic linings, we should consider the femoral head, and after that, again, the strength of the total stem. And these calculations, the ratio of the materials, the selection of the materials matters a lot. Then comes a challenge. How to select the material? Is there any blueprint of selecting the material? Is it just hit and trial method of the selection of the material? Are there any questionnaire to selection of the material? Or just seeing that someone is using alumina, let us use alumina. If someone is using, different metals in different ratio, let us use that. How to develop the critical thought process about selection of the material? I tried to keep it in a single slide in next few slides it will come. So selection of the materials are the major challenge, and once we will have master on those how to select the material, if we will ask the question from ourselves, what should be the property? Because we know the biology part, we know the material part. So we should ask the question, being a biology person or being a material person, and the material which suits all of them will be the best material to develop the biomaterial for any of the bone. बाबू चुप हो जाओ, पढ़ा रहे हैं ना. Sorry. Now, if we consider the normal hip or the arthritic hip, we all know that the space between the femur and acetabulum in the normal hip. But in arthritic hip, the space is lesser or even no space. Since the cartilage is missing in arthritis. So what should be the like how to solve these challenges? If we are focusing to fill the gap with the porous material, then again we are actually solving half of the challenge. We can have a biocement of PMMA, which will adhere here, and we can fill it up. But if we are thinking to provide it with the porous coating, then we are almost solving the challenges by mimicking the space of that particular, not like if we consider the normal hip as well as arthritic hip, then we can actually biomimic this space. So biomimicking is again a different concept because we have to mimic the environment over there. We should think about the compatibility, we should think about the durability, we should think about the degradability, we should think about the infection, we should think about the immune response, we should think about the patient compliance, we should think about the mimicking the environment over there. Now, like being a bioengineer, where should I look? Like if I'm starting as a biological person to work in a biomaterial perspective, where should I start? I'll just share how I have started. If we just see the yield strength as well as the Young's modulus of different materials, we have the elastomers, which will have the elastic property. We have the polymers, and we have the biological materials. In these biological materials, we have the calcareous bone, like of low density, of high density. So we need to check out where our material lying. And if we will have a critical study of this particular image, then we will be able to understand which material we should use for low density, means for low density cancellous bone, or for high density. Which material we should use for cortical bone? Which material we should use if we need to develop the bioglass for some function? Where we should use the metals or alloys? We have heard about nitinols in many of the case studies. Where we should use the ceramics. So fundamentally, if we speak, we should understand the subset of the biological materials. Why I'm giving it a term of biological materials? Because they are biocompatible. They are being not rejected with the body in terms of immune response. So if we will select some of the materials which will have the functionality of or the matching property with the biological materials, then we can meet our means we can meet the requirement of the biomimicking. We may have the composites, we can add the ceramics to give it the different mechanical property. We can have metal alloys and ceramics because these are the stiffer. So if we need the stiffness, we can add them. So if we consider the material selection, Professor Mike Ashby and co-workers in Cambridge, they have given some of the flowchart in this particular book. If we need, let me introduce the book first, because if we need the design-led book, then the materials, and if we want to go to the advanced level, material selection in mechanical design. So, being a biomaterial, we need to learn the material selection process. These two books might not have discussed, actually it has not discussed with respect to the biology. Few contents have been discussed with respect to the biology, but if we will refer the capsule of this book, it will give us a thought process for the material selection. Like we have all the materials. Then we need to understand what is the function. Whatever the component, the biological component or the mechanical component inside the body, over the body, for the biometrical material property, we want to develop what is the function. A wound healing patch has different function. A drug delivery vesicle has different function. A compact material for bone has different function. A valve biomaterial is different function. A hernia mesh has different function. Based on the function, they have different constraints. What essential conditions must be met? A hernia mesh should be flexible and should not break. This is the very fundamental mechanical condition which we require. What objectives we have? What should be maximized? What should be minimized? Because if we are going for bone-based biomaterial, then it should not have the flexibility mainly for the hard bones. We should need to screen based on the constraints if we need to go or no go for these two materials. Means we should think about the function constraints and objective. We should rank based on the objective. Screening will go with the constraint, what essential condition must be met. And after shortlisting the material, we should rank the materials based on objectives. Let's say if we are talking about, bone implant or the filling the gap with the biocement. If we will fill the gap with metal, it might not justify. So in that case, the ceramic material will be the best fit for those biocements. So we should rank the biomaterials based on the objective. We should screen the biomaterials based on the constraints. And the top candidate materials, let us say if we have 3 materials to be selected as a top candidate, we need to go for supporting information. Maybe the databases, maybe publications, the patents. After that, we need to analyze it based on the local conditions, that whether we have the supplier. Because I can remember, like we were discussing with Reliance and they were trying to develop the material based on some very ultra high molecular weight polymer. I'll not name the polymer. But what we realized, that time I was in industry, and what we realized, whatever the systems we have, the local conditions we have, we might not be optimize the recipe to further take ahead. So obviously the supplier should be the local one. We should think whether we have the machinery to optimize the process or to even optimize the recipe of the polymer, the location. So local conditions also is actually very important to understand us. In our case, our lab condition is actually very important to understand us. And ultimately, we were bound to say no to Reliance for that ultra high molecular weight polymer because we were not able to handle it. If the material satisfies all the criteria, then it comes about the biological study. Being a biomaterial person, because my basic is like I have handled the stem cells, even a cell line. So being a biomaterial person, what I realize, if I can use Hindi, हमारा material तो बन जाएगा, हम लोग बनाएंगे और bio तो हो जाएगा. Bio is not a challenge for us because we have very specialized scientists. Many of the concepts. Let me cite here the discussion which I had at Lucknow with Navedya sir. Navidya sir is very recognized biomaterial scientist. He has very much good command on bone, which I don't have. But I have command on formulation. Whatever the challenges he was facing that time, We have just enlisted. Being a material, we came to the three solutions. Those three projects are still with me, and like obviously we will submit. But yeah, why I'm citing this? Because a biology guy, the biological person who has worked years and years on a concept, knows the challenges. Being a material, we have to match each other and we have to solve the purpose. Biology is not a challenge. We have like we can collaborate with the experts. Selection of material is a challenge because that need a tremendous questioning, which should satisfy the biological person. Even I remember in our discussion, two of the concept whatever I have given, which are very naive, Navija sir has rejected it. Straight ahead, he said no, because he know the biology. So coming to the point, selection of material is actually very much like a bigger challenge. अगर ये हो गया तो फिर तो ये हो जाएगा बायोलॉजी हो जाएगा. So most of our advanced case studies are systematic methodology. We need to learn about the material selection first. This is what I believe. And here again is a challenge. Like a mechanical engineer might not have that much interest to learn about the biology. Similar is the case, a biology guy might not have much interest to learn the mechanical part. But we should develop it because material is the actual in coming years, materials have the era because if you will see the patents trend, most of the patent are going on the formulation. Either they are enhancing the process or they are like making the ease, or they are going for the new formulation for the same task in any domain. So material is the emerging domain, which and the biology guy, if they will develop their skills in material selection, they can do tremendously good work. It is based on the translation of the selection of the task and function constraints and objective, which we have discussed earlier. The screening material candidate on the constraint, we should remove the unsuitable material. We should go for the suitable material, whatever we have ranked. And the first ranking to the candidate means the material candidate, it should be given on performance index. And after having shortlisted material, we should discuss with the biology person and then we should rank it, because the real objective is there with the biology guy. It's not with the material person. So I'm sorry. Something has happened. I'm sorry, sir. Just give me 2 minutes, sir.
Sure, sure, sure.
Just 2 minutes. Something has happened.
You have difficulty in projecting the slides?
No, sir, I don't know. Suddenly my slide has got closed, and now I'm just looking. I'm just opening the slide again.
Okay.
Maybe my laptop issue because I'm not in my office and I'm just using my father's laptop now.
Want to upload it again?
Yeah. So I think major challenges are visible now.
Yes, 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 visible. Visible.
Yeah.
Please go ahead.
So whatever the learning I had being a engineer or being a biotech person, the major challenges as I'm focusing throughout my presentation is the material selection, because we should know the chemical composition, we should know the blending, we should know the copolymerization, we should know the functionalization, we should know the polymer molecular weight, we should know the molecular mass, we should know the polymer solvent solubility, etc. We should know the solution variable, how the solution or how the composite, even how the mixture will behave, whether it will interact with each other or not. If they are interacting, what the final formulation will be. We should know the process variable, how to optimize the recipe for any process, because if we are developing a material which involves more than one material, then there should be a process. Even for dissolving some polymer, there is a process. So we should know the recipe. We should know the, if we are encapsulating any drug in the biomaterial, we should know the wall is to core encapsulation and those physics behind it, the chemistry behind it, and the biology why we are doing this. We should know the encapsulation efficiency. We should know the biomaterial stability. We should know the final action, how the biomaterial behave at the site. We should know if they are degrading or not. If we talk about the strategies for material selection, we should again know why we are developing, what are the biofunctional perspective. If we are working on drug delivery, or if we are incorporating the drug into our biomaterial, whether the drug is interacting with the biomolecule or not. Let me again share the story which I was working with the Dr. Reddy's. They were telling that the like edible polymer should encapsulate the drug, and it should deliver the drug through oral route, whatever the polymers we have opted for. So the polymer was itself getting reacted with the drug after encapsulation, and it was changing the crystal structure of the drug, not chemically, just the crystal structure. Those like change in the crystal structure, the final bioactivity got manipulated. So the polymer, the material, if we are encapsulating the drug, it should be inert enough. It should be biocompatibility, bioavailability, all these things. Again, the final thing, whatever the formulation, it should have user compliance. The FDA and regulatory authority should approve the material. Here, very means, take home message is this. I have learned from several failure. The take home message for students are, if you are using any of the formulation in your biomaterial development, so the material, try to choose those materials which are already FDA approved, or you will not face challenges in taking the regulatory approval. It means it should not have the adverse impact. Again, this is again a challenge, means it should have reproducibility. If it is like we all know the European culture, they do perform the same experiments in several labs all together, and these labs should have the similar results. So it should have reproducibility, otherwise technology transfer will again be a challenge in terms of biomaterials. It should have possible large scale production. If we are optimizing any of the formulation, any of the recipe, we should keep in mind that in Indian scenario, or for whatever the industries we are working on, it will consider the scaling up parameters we should consider. Again, the major challenge which I still face is a proper characterization. See, characterizing the material, if it is not with the biological domain, means not in the biological environment, is quite easy. We can go to SEM, TEM, we can go to whatever the characterization instrumentation available. But once we have given the biomaterial, implanted the biomaterial, or we have given the material in drug formulation, how to characterize it? Even forget about if we have implanted. The major challenge which I still face is if we have encapsulated the drug. into any formulation and the formulation is nano-sized, characterizing the size is quite easier. But how to characterize the drug inside the encapsulation? These are the challenges, and that time chromatography is the best friend. Again, we have to dissolve it and we have to characterize it, but we should have command on that. After that, surface morphology and analysis, unless and until the material is not inside the body, characterization is quite easier. So we have to plan. What are means limitations which I still face in many of the formulation is, the toxic behavior of the new formulation. If I want the localized on-site or on-response behavior, let us say like with Navidya sir itself for joint formulation, we were trying to develop some gels. And based on the weight of the body, this is the signal. I'll not go deeper into the formulation because we have not submitted the proposal yet. So based on the weight of the body, some signal should be there into the formulation so that whatever the biofunctionality we require, it should meet the criteria. So these are the challenges. Discussion among the biology guy and the biomaterial person is actually required. After that, desired pharmacokinetics and the dynamics, analyzing the material over any of the biological model, right? Small animals, it's easy based on the functionality. If we have wound, we have given the wound healing patches. Fine, our wound got healed. But actually going to the molecular level, how the material has behaved on site, that characterization protocol is still not yet optimized for, like, different lab do follow different protocols. So biocompatibility, bioavailability, and effectiveness is still a limitation for us. Altering the pharmacokinetics and extending the drug half-life, targeted drug delivery. Apart from that, lowering the drug dose means what I mean. When I was developing the material, means a biomaterial coated, single-sided, so that post-operational contamination can be avoided. Why we were coating that like on the biomaterial, so that the drug loading means on the body or the drug stress can be minimized. Fine. But what about the stability? Stability, unless and until the material is not going on site or it is not being implanted, the stability will be good. Fine. How it will behave in the biological system? Then again, we have to create a simulated microenvironment to analyze those behavior. And simulating the or mimicking the microenvironment is again a challenge. It might vary from patient to patient because patient X is having different food habit, having different lifestyle. Patient Y is having different food habit, different lifestyle in all the terms. Obviously, the microenvironment in our biological fluid also varies. And how these two biological fluids will interact in different scenario, it is not yet optimized. As far as I know, it is not yet. Mitigating metabolism and drug-drug interaction, drug biomaterial interaction, biomaterial and biological system interaction. From one organism means from one patient to different patient, how the biomaterial behave. These are the actual limitations. But again, we have to go with the majority. So yeah, and finally we have to obviously reduce the toxicity. So this is what I have shared, whatever the learning I had till now from my industrial experience and yeah, discussion with the experts like Navidya sir. So that's all from my end.
Okay, thank you very much, Shivendu, for a very lucid overview of a biomaterials perspective for bioengineering. Um, I'm sure it would be very much useful for the beginners, no doubt about it. Um, some again, you mentioned it several times that you had a discussion with me, and me means from the perspective of a bone biologist. So I'm definitely not a bioengineer like you, and particularly issues related to what we call issues related to drug delivery. Now, um, delivery in what we understand as a layman in the drug delivery is that bone, it is a highly vascularized organ, and delivery is um, feasible. But what about cartilage? That part we don't know. And if you are considering delivery to joint region, what is the state of the art that is going on now?
So as I mentioned that I'm not a bone biologist, but again, from our discussion and whatever the readings I had, so if we consider the joint region, delivery to block the infection till the post-operative chances of contamination are required, it might be possible if we have the sustained delivery. Let us say if it can, the sustainability can be for 6 months. But again, that 6 months sustainability will be a challenge because we cannot dissolve the drug just to like, because we have to biomimic the joint region fluid as well. So dissolving the drug might change the behavior. In very layman terms, it will affect the viscosity. It will affect the lubrication property of the fluid. So yeah, but we need to look into the sustainability until it is means whatever the time period it is required post operative.
Actually, maybe I was not absolutely clear in my question. By targeting the joint or the cartilage, I meant by not local application, but is it possible if you have to do it by the systemic route? So, if you are trying to do it, trying to target cartilage from
Mhm.
IV route, is it at all thinkable at this stage?
I will
I mean,
being a like
may not be
I'm
thinkable at
like
this
I'm
stage.
not able to answer because again we
Okay.
I need to go to the biology part.
Okay, okay. No worry. Now, generally, for particularly for the sake of the beginners, you have quite extensively talked about the materials that are being used routinely for biomaterial that are non-toxic, and you also mentioned towards the end that are generally approved by the regulators for human use. So most probably you are relying very heavily on things such as PLGA or PEG or alginate kind of things.
So there is one database. ANSYS company is actually has developed the database,
Oh, is
and
that
they
right?
have
Okay. Ah,
yeah. So
okay.
in that they have developed the level one, level two, level three, level four type of biomaterials, and
Right.
and if we will go through it, they have categorized it. but that is also based on the data, whatever the available through patents and publications.
Mm-hmm.
So while selecting, let us say, if we want to go for the biocompatible, they have the list of the biocompatible, materials. Again, in that, if we go for the stiffer material, they have the list of the stiffer biocompatible materials. So we can shortlist it, and at the end, we will come to the 5 or 6 materials, and then we should then we should discuss with the biological guy that these are the material, these are the property, these are the formulations we can change. What are the challenges are you facing? If it will solve.
Okay. Okay. Now, when you are talking about the delivery systems in terms of the formulation that you do, are you what kind of expertise you have in terms of the delivery system? Are those basically the nano
So
droplets
till
or
now, till
liposome?
now, the delivery, whatever we have developed, means my team has developed. It is nanofiber based encapsulation. It is nano emulsion based encapsulation of the drug. It is liposomal, nanoliposomal form of
Nano.
drug delivery. We have proposed aptamer functionalized for
Ah.
targeted drug delivery in TSC. That the project I have discussed with you also.
Mm-hmm.
Uh, Bharat Biotech was the collaborator. It
Right.
has been shortlisted. We have given the we have presented our part, but the decision has not yet come. But frankly speaking, till now I have not gone for targeted drug delivery because means maybe I just entered into the material part more. Yeah. So now I'm slowly going into the targeted or response of specific drug delivery system. But yeah, slowly I'm entering into the field.
Now, I have come across from you discussing with you things such as, if I remember, supermagnetic, paramagnetic, responsive
Piezoelectric.
nanofibrous scaffold means. What are those?
I was not means frankly speaking, I'm just young researcher, so I was just skipping to name them. Yeah,
Okay,
but yeah, those were the response specific, like let us say piezoelectricity. We have piezo one protein in our fluid,
yeah,
mainly near by the joint, and mimicking that will be possible using the piezoelectric polymers. We
okay,
have different piezoelectric polymers which we can use. We have silk is one of the most biocompatible and piezoelectric polymer. So those polymers we can shortlist and we can have a gel formulation. So and piezo means once we will mimic the piezoelectricity over the joint, the chances of like cartilage regeneration will be possible. So yeah, in simple term, the biomimicking, it reflects everything. Means it should mimic the even piezoelectricity, the magnetic property of the material, of the requirement on site. So this is what, in a sentence, biomimicking the environment is actually required.
yeah. I just wanted to impress upon the students and postdocs is that the challenge is so immense, particularly even if you think about just think about regenerating cartilage. It itself is a massive challenge for the entire world scientists working in this area, and then to mimic the joint in terms of its other physical and physicochemical property, which is planning to do as such is are um, quite quite challenging, if not extremely ambitious. But unless you have those kind of ambitious goals, you cannot really think of achieving it. I will now stop and let others ask question. If anybody has any question, clarification, or if you have not able to understand some issue, just feel free to ask Shivendu. You can raise your hand if you want to ask. Looks like there is no further question or clarification. So in that case, I will not hold back Shivendu. There is one. Please, Anupam, ask.
Hello, sir. Good afternoon, everyone. I have one question, sir. In the slide where you talking about the hip implant or femoral head implantation, there was a sequence where wire resistance are decreasing quite a bit by using from ultra high density polyethylene to the aluminum. And when the next slide it's come up with a porous structure for bones where you call said the bone cement with porous structure, the biocompatibility issue is solving by using the porous structure. It is not possible that we have to sacrifice the wire resistance. The wire resistance will be more if
नहीं the
we
porous अच्छा yeah you
when we taking the porous structure, the wire resistant will sacrifice.
I understand your question, and porosity always should not get confused with the solid state porosity.
Okay.
Means the adsorption property we can give it to the semi-solid also. So if we will use the semi-solid structure, we can mimic the environment over there.
Yes, sir. Thank you.
By the way, sir, Anupam is my own PhD student.
Oh, okay. Okay, okay, okay. Okay, so any other question? Raise your hand, please. Doesn't look like any further question. In that case, I would again thank Shivendu, and I wish you all the best for your very exciting ideas, and I hope they translate and humankind and particularly patients are benefited from your research. Thank you very much.
Thank you so much, sir.
Bye.